It's a little bit slow. Okay. I guess it will work. Let's hope it will work for our fellows abroad. Okay, so let's start today. I will tell you something about the innate immune system. I think it really matches what you have been or should have been studying for the case of today, so that's nice. If there are any questions, just ask them while I talk, and if anything is unclear, also if you would like to know more, or if I go too fast or too slow, or if well, just interrupt me, no problem. So I'm going to talk about the innate immune system. My name is Sander Rensen. I'm working at the Department of Surgery, and in this department we do, of course, a lot of surgery, and basically when you yeah, it's quite obvious, but it's true. And this also involves activation usually of the innate immune system, because you induce a wound, and you know that for a wound to heal, a wound healing response needs to be started, and this always involves the innate immune system. So we've been studying the innate immune system quite a lot, and I myself have been working mostly on obesity and the metabolic syndrome. And while you maybe think that this is, well, not really related to the innate immune system, over the last years it has become clear that also in these fields of research the innate immune system is really important, and I will sometimes touch on it a bit in the presentation. So just a brief outline for what I will be talking about. First, a very broad general part about what the innate immune system is or what it comprises. Then the elements of the innate immune system that you can distinguish. So the barriers that are part of the innate immune system, the different cell types that are part of the innate immune system, and also various proteins that really belong to the innate immune system. And I will focus on the complement system, which I think you also have been studying, right? Yes. So that's good, because sometimes students think it's complicated. So if anything is unclear just about that, I know a lot about it. So just ask. And then I will go into more detail a bit about the molecular mechanisms that are involved. We will talk about how the innate immune system recognizes danger. It uses, for example, toll-like receptors, but also, again, these complement factors that are here. I will talk about the process of phagocytosis, and also about some cytokines and the actions that they execute. So, very briefly in general, it's not so easy to use the pointer here, I think. Well, I can try, but usually we just go from above to below, so it should be clear. So the innate immune system is a very old system, and therefore if you study any organism, doesn't really matter which organism, there's always something of innate immunity present, and that's why we can use for research a lot of model organisms to study the innate immune mechanisms. Basically in all multicellular organs you encounter some kind of innate immune system, and that goes all the way up to the sea urchin. And all different kinds of fishes and amphibians, and they all have this innate immune system very much developed usually. This innate immune system is you can view as the first line of defense, and it provides an immediate protection. So it's always there, it's always present, and it's always ready to go, so to say. And it provides protection not against a specific organism or a specific danger, but against a really big variety of different dangers, and it can be pathogens, but it can also be malignant cells that have to be removed, or other injury-related debris, for example. So this is why the innate immune system is sometimes, or many times actually, referred to as the non-specific immune system. Also, while the adaptive immune system has a memory response, when you get antibodies and T cells that are specific for antigens, there's always also memory cells that are formed that are important. This is not the case for the innate immune system. So this really is important for the first exposure to dangerous pathogens mostly, and also transformed cells. And it's already effective in children, although with my kids I'm not always sure that it works that effectively. But it should be there. It should give immediate protection, and it's already present at birth. Not fully developed, but most of the elements are already active. As I said, most of you maybe think of the immune system, the innate immune system, as to protect us from pathogens, but it's equally important for the elimination of cells that have been damaged or that have been transformed, that have been infected by viruses, of course also, but also when you get just a physical injury to cells, it's also important that this is recognized and removed by the innate immune system. And because it's so important, defects in the innate immune system are quite rare. If you have a serious defect, usually it's not compatible with life. So you don't encounter a lot of real immunodeficiencies in the innate immune system. So I told you that I've been working on the metabolic syndrome, and just as just an overview of why innate immunity is also important in this field. Because, for example, if you look at type 2 diabetes, then in the adipose tissue of subjects with type 2 diabetes, there's a lot of inflammation going on actually, and most of this is characterized by an innate immune response. And this inflammation of adipose tissue really is an important factor in driving the diabetes in these subjects. And the same holds actually for the insulin resistance, that is more or less a pre-stage of the type 2 diabetes, of course, which also involves inflammation in the skeletal muscle. And in the liver of obese subjects, you also see a lot of inflammation usually, which is related to the fat accumulation in this liver of these subjects. And this also is characterized by an innate immune response. And the same goes for atherosclerosis, which is also these plaques. They consist mostly of cells of the innate immune system. So it's not only about infection and injury, but also about this kind of diseases that the innate immune system is important. Then, if you think of what the innate immune system should protect us from, it's very diverse. It has to protect us from viruses, which are very small, bacteria, which are still small, but a little bit bigger, of course, in a very different nature. Fungi and yeast, parasites, and as I said, also own cells that have been altered or damaged. These all should be cleared by the innate immune system, and this is a very difficult task, of course. And you can see later on that there's lots of mechanisms and cells and proteins that take care of this. And also, if you think that some dangers are present in the outside world, so to say, extracellularly, and others are present intracellularly, this is also, of course, very different for this kind of system to protect us, because the proteins need to be active both extracellularly and intracellularly. And also, if you think of the dangers, that the size of the dangers really also is very different. Tapeworms can go up to 25 meters. These also should be effectively cleared by the innate immune system. And parvoviruses are very small. These should also be seen and cleared. So this is very difficult. And that's why the innate immune response is very robust and also redundant. This means that there are many different lines of defense for every attack. So it's just not dependent on one mechanism or one effector cell, but there's many systems that cooperate and that go together and work together. And if one of the elements is compromised, then there's still another element in place which can take over. So if we dive a bit further into this first line of defense, then the first thing that we encounter, and the most obvious barrier, is really the physical barrier that you can see at basically all interfaces between the outside and the inside world. So of course the whole gastrointestinal system, there's a barrier from the mouth to the anus. The skin is a very important barrier, a place where an innate immune barrier is present. with the eye, also then the urogenital tract, and also the respiratory tract. So basically all the places where there is interaction between the outside and the inside world, there's a really important efficient barrier present which belongs to the innate immune system. And this can be further divided into three parts. So the first part is the physical barrier, which basically really consists of epithelial cells mostly, but also hair is part of this barrier, and also mucus that is present, of course, at lots of mucosal interfaces. There's a mucus barrier which is also really important. Then there is a so-called chemical barrier. These are the compounds that are present in, for example, sweat and tears and saliva, but also other body fluids. And for example, you can also view the low pH in the stomach as a chemical barrier, because it also gives a protection against infection. And then the third element is the so-called physiological barrier, which is comprised by our normal commensal microflora. So these bacteria that are present in our gut, but also on our skin, these also form a kind of barrier against more pathogenic bacteria. So if we look a bit further into the epithelial barrier of the skin, then we can say that this is the most difficult barrier for microorganisms to penetrate. And this is because there are a lot of layers of cells present, so they are all on top of each other. And of course, if there's many layers, it's harder to breach this barrier. And furthermore, the cells that are on the outside, so the outermost cells, are embedded with keratin, which is an intermediate filament protein. And this is a really a structural protein which gives a lot of extra rigidity to the barrier. Furthermore, as you know probably, the skin is very frequently renewed from the lower layers. And in this process, the outer layers are always released and sloughed off, and all the microbes that are present on this outer layer are also discarded in this way. And then there's a lot of, so on top of all these things, there's also a lot of antimicrobial proteins that are being produced in the skin. And this is part, of course, of the chemical barrier in the previous slides. And there's, you can see here a whole list of different proteins that are all have all antimicrobial actions, and as you can see, they are synthesized in different parts of the skin, and so there's a real cocktail of very dangerous molecules for organisms that would like to penetrate our skin. So this is the epithelial barrier of the skin. So if you look at the epithelial barrier of the intestine, you can see that it's very much different. In contrast to the skin, here there's only one cell layer that separates the outside world from the inside world. So it's really maybe much more vulnerable, you could say. So if you look here at the intestine, then these cells here, these are the epithelial cells that you know you've been studying before as well here. And this is the lumen, so and this is the inside and this is the outside world. It's just one cell layer separating it, and this means that the cell, the barrier is differently structured. And in the intestine, it's really the tight junctions that have been very much developed. So the junctions that tie the cells together and that make sure that there's a good seal. These are really strong and tight in the intestine. And then there's another important part of the barrier, which is the mucus, which is present in the intestine. This mucus, it traps the microbes, and this mucus consists of glycoproteins, so proteins that have a very high molecular weight. They are very heavily glycosylated, and because of this glycosylation, they attract a lot of water, and this is why this is such a viscous layer. And these proteins are mostly produced by goblet cells. And well, if you look here at, maybe I can point it out here, it's easier, I guess. So if you look here, this is the epithelial cell layer. with here the goblet cells, and these are cells, for example, that secrete antimicrobial proteins. We'll talk about it a bit later. But the goblet cells here, they make the mucus layer, and if you look in the colon, you can see that there's a very big mucus layer. Here it's shown. In the stomach it's also quite big. This is to protect the cells from the attack from the hydrochloric acid, which is formed there, which of course is very aggressive to the cells. In the small intestine there's a little bit less mucus, but here in the colon there's a lot of mucus, and of course this is also related to the very high concentration of bacteria that is present, especially in the colon, but less so in the small intestine. So in the colon there's a really nice organization of this mucus layer. There's a loose outer layer which is very big, and in this outer layer there are microorganisms. It's always the risk. This is the outer layer where there are pathogens or other microorganisms, not necessarily pathogens, but microorganisms can be present there. Then there's close to the cells there's a really firm and dense mucus layer which basically prohibits entrance of microorganisms. So this is more or less a sterile environment, and you can see that there's lots of antimicrobial proteins present, also dispersed in the mucus, and also antibodies are present in this mucus layer. And together these elements make sure that microorganisms cannot enter the system. Then there's always, or in the intestine at least, and also other systems, there's sometimes a peristalsis involved, so a flow, which of course also removes bacteria from the body. So again here, this is just to show the differences between the intestine and the skin. Skin, a very efficient, difficult layer to break. Many cells, many antimicrobial proteins. These are also present here in the mucus in the intestine, but just one cell layer, and this is really the big difference between these two epithelial barriers. Then if we look further into this chemical barrier and the different kinds of antibacterial proteins that can be generated and that can be present, it's clear that these can be made by many cells, but mostly these types of proteins, they originate from epithelial cells or from immune cells. And basically almost all epithelial cells can synthesize these antibacterial proteins. In the skin, in the intestine, we have seen it before, but also in the lungs and the urogenital system, salivary glands, tear glands, they all are capable of making these antibacterial substances. And of course the immune cells and mostly monocytes and macrophages and neutrophils, they are also really efficient and make a lot of these antibacterial products. And important examples in this respect are lysozyme. And lysozyme is an enzyme which degrades peptidoglycan, and this is a part of the cell wall of both gram-positive and gram-negative bacteria. And so this enzyme is capable of really making holes in bacterial cell membranes, and in this way it kills bacteria. And it's most effective in killing gram-positive bacteria because they have lots of peptidoglycan, and gram-negative bacteria are also protected next to the peptidoglycan by another cell wall which mostly consists of lipopolysaccharides, which we will see later on. This enzyme has really high concentrations in tear fluid, in saliva. It's also just present in the blood and also inside the immune cells. Then another important example are enzymes which are called peroxidases. These enzymes, they break down hydrogen peroxide, and in doing so they produce a lot of very reactive oxygen species, so oxidizing agents that basically react with everything they encounter which is close, and in this way they really damage anything that is close. and also if this is an microorganism, they can effectively eliminate or kill such a pathogen. And high concentrations of this enzyme, these enzymes are found in saliva and also again in immune cells. Another example, another class of these antibacterial substances is made up by lactoferrin. Lactoferrin is a protein which also has a very high concentration in blood but also in saliva. And this protein has a very high affinity for iron. And iron is important for microorganisms to proliferate. So if this is sequestered, this iron by the lactoferrin, then it's not available anymore for the pathogens or the other bacteria, and in this way their growth is limited and inhibited. So this is another type of antimicrobial protein. And then the last class I would like to name are the defensins. And these defensins are also antimicrobial peptides. They are very small and mostly made by immune cells, but also by epithelial cells. So for example in the intestine, and there are specialized cells, the Paneth cells, which are in the crypts, which make lots of these defensin proteins. But also the enterocytes themselves are capable of making defensins. And these peptides also integrate into the membrane of microorganisms and in this way form pores in their membranes and kill them. So this is different different examples of antibacterial proteins, which are also part of the innate immune system and not maybe always, how do you say, you don't read a lot about them in the textbooks, but are also important elements. And just how important I'd like to show with an example. So these are our two experiments. On the top you can see an experiment with mice in which they made mice that express a lot of lysozyme, so the so-called lysozyme transgenics. They compared them with normal wild-type mice in this case, and they also compared them with mice that lack lysozyme, so that cannot make any lysozyme. And all these groups of mice had been infected in their lungs. And you can see, this is a survival curve, that most of the mice that make a lot of lysozyme, with the squares here, they can handle this infection and they survive the infection. Whereas wild-type mice, well, they are not so resistant, and some of them survive, but most of them die actually. And if the mice lack lysozyme completely, they cannot cope with this infection and they all die. So this single molecule really makes a big difference to resist this challenge. And this is the histology that goes together with it. So you can see lots of inflammation after 24 hours in all groups. But you can see also that the wild-type mice and the lysozyme mice, they can handle the infection. There's still a structured lung tissue present here, whereas in the mice that completely lack the lysozyme, you can basically do not recognize any real lung structure anymore here. And the same kind of phenomenon you can see in for defensins. So this HD5, this abbreviation stands for human defensin 5. And human defensin 5 is very important to resist salmonella infections. So again, here they took mice that expressed a lot of defensin 5, or just wild-type mice, and they infected them orally with salmonella. And the transgenic mice, they don't care, they just live on happily ever after. But the wild-type mice, you can see here, after 2 days, most of them, or actually all of them, died, and they cannot survive this challenge. And just by giving them extra defensin, they can handle it. So, just to show you the power of these proteins. Then I've been talking about this physiological barrier here, the third part. So we have seen that there's a chemical barrier, a physical barrier, and now we will talk about the physiological barrier, which you can view as the normal microflora that are present in our body and on our body. Basically, of course, they are not really part of our immune system, but you can see them as a part of the first line of defense. And this, they provide really quite a lot of protection, and this is because they exclude invading microbes. So these microflora that we carry with us, is usually harmless, so it doesn't do any harm. Sometimes it's also advantageous. These bacteria can maybe make vitamins for us, or produce extra energy from the diet that we take. And they also, by just being there, they provide a kind of competitive exclusion for microbes that should not be there and that try to invade. And both, you can see that they compete for space, but also, of course, for nutrients that are present. Then these microflora, these commensals, they also make sometimes substances that are toxic to other bacteria that might be pathogenic. So in this way, they also provide protection. And another way in which they provide protection is by stimulating our immune system, and you can see it as a kind of exercise. So they always keep our immune system alert, because sometimes, of course, they can also become opportunistic, as they say, and these in principle harmless bacteria can become harmful, but our immune system usually can deal with it very well, and in this way it's really being taught to do so. And again, just an example of how important this part of the barrier is if you look at germ-free mice, so mice that do not have any bacteria present in their gut, but also not on their skin and other organs, you can see that this really affects their function quite a lot. They have a really big reduction in the size of all lymphoid organs. They have low antibody levels, antibody titers, and a very hampered and little immune response. So this is really obvious. And I told you before that innate immunity and the aspects that are going together with that also are related to body weight gain and metabolism. And you can also see that these germ-free mice, they also are very resistant in gaining weight. So you can feed them a lot of high-fat diets, for example, but they don't gain easily weight if they don't have their normal bacteria present. So the barrier is quite strong, but still, of course, it's being broken a lot. You all know about it. We all have been sick before, unfortunately, and this usually means that one of those barriers has been compromised. And you can see, for example, especially in the respiratory system, but also in the gastrointestinal system, lots of these breaches occur. And if the barrier is broken, then bacteria, microbes, they can enter. And if they have entrance to the tissues, they usually can also spread, because they can, of course, use our blood vessels and lymph vessels to really spread. And this, of course, you want to prevent. And you want to contain a possible inflammation or a possible infection. And in this sense, the second line of defense comes into play. And these are the so-called innate immune cells. So, and this is an overview of the different cell types that are present in the innate immune system, that belong to the innate immune system. On the left, on the right, these are the adaptive immune cells. As you can see here again, of course, this innate immunity, as I told you before, gives a rapid response, so a really rapid action. And this is mainly due to the actions of macrophages, of natural killer cells, and especially also neutrophils. Then there are eosinophils and basophils, which are also classified with the innate immune system. Mast cells are present, and then the dendritic cells. And of course, complement proteins are not cells, but they're also an important component of innate immunity. And well, I will talk about the granulocytes later. Then there's a couple of cell types that are not so easily categorized, so they have characteristics of both the innate and the adaptive immune system, natural killer T cells, gamma delta T cells, and of course there's the classical adaptive immune cells, the B cells and the T cells, which you can further divide, but which will be covered later in the block. Then looking a bit more into detail about the cells of the innate immune system. A very important cell type is the neutrophil. You've been studying it before. It has this characteristic shape of the nucleus, segmented, lots of granules inside its cytoplasm, and this cell is really active in performing phagocytosis. It can also make a lot of reactive oxygen and nitrogen species, which are also important for protecting. And it, as I said before, it can also make a lot of antimicrobial proteins and peptides. Then macrophages. They're also very active in phagocytosis. Come into play later. I will talk about it in detail in a minute. They make a lot of inflammatory mediators such as cytokines and chemokines, but they also have this antigen presentation function, which is really lacking from neutrophils. So neutrophils, if they have phagocytosed something, they do not present it to cell types of the adaptive immune system, whereas macrophages they do. So this is an important difference between the two cell types. They also secrete, like the neutrophils, lots of oxygen species, reactive oxygen species, nitrogen species. And they are also important for bringing to a site of infection complement proteins, which are also important, as I will tell you later. Then these dendritic cells, I just want to touch on them briefly here. They are very important for antigen presentation, so they can also phagocytose particles or cells or pathogens, and they have a really specialized function for antigen presentation. To do that efficiently, they also need a lot of co-stimulatory signals, which they also present to the other cell types. They can also make reactive oxygen species and cytokines, but this is a different type of cytokines as compared to the macrophages. And then natural killer cells, also very important, especially for the lysis of cells that have been infected by viruses. Interferon is a cytokine which is really important for this function of these cells. And they can also, when they have cleared a pathogen, they are also make sure that macrophages are activated to clear up the area and to clear the tissue debris. Is everything clear? I will talk about it, not specifically about these interaction between these two cell types later, but they mostly just use cytokines really to communicate. So cytokine is secreted by one of the cell types, and cytokine receptors are present on the other cell type, and in this way they can interact and communicate, also quite distantly from each other. And then other cell types, mast cells, eosinophils and basophils. Just briefly, mast cells, they are cells that are resident in tissues. So neutrophils are mostly circulating and only in tissues, are only present in tissues when there is an infection. Macrophages can be present, also resident in tissues. They are not present in the system, in the circulation, because then they are still in the form of monocytes. But mast cells are usually resident in tissues, and they play a role in allergy and wound healing. And this is because they contain a lot of granules with histamine and heparin. And these proteins have a role in these processes. And mast cells are also important for the defense against worms. Eosinophils, they are it's a little bit unclear exactly still what their function is, but they appear to play a role in the defense against viral infections, just like the natural killer cells. And they also are important for clearing multicellular parasites, like worms, for example, also again. And they're also involved in allergies, so there's lots of similarities between mast cells and eosinophils. But the real specific function of these cells is not so clear yet, still not. Just like with the basophils, they also contain granules with histamine and also enzymes that perform hydrolysis, so can facilitate a lot of reactions. But the exact function of these cells still also is unclear. So, this is so neutrophils, macrophages, and dendritic cells. They have really this defined capability of performing phagocytosis, and this is why they are also sometimes referred to as phagocytes. So these cell types can do that. So what? Yes. How does heparin? Heparin has an important role in clotting, so blood clotting, and by interfering with that process, it can alter the wound healing response. So it's really more a function in the wound healing cascade than just really in a protection against pathogens. But how do you say, to release these proteins, lots of times heparin is co-secreted with histamine, for example, and they're sometimes in the same granules, and sometimes they are released in the same time, so they have a different function, but are co-secreted, so to say. So these phagocytes, these cells are really specialized cells. They have a very specialized function. They engulf and digest pathogens, and also cellular debris. So when a cell has been injured, damaged, or transformed, of course, the cell parts should also be removed. This is also done by phagocytosis. And also the normal cell turnover. So if you look in a tissue, of course, there's always renewal of cell types going on. And the old cells are always removed by a process called phagocytosis. This is performed by phagocytes usually, and these phagocytes can be alerted to signs of microbial invasion and tissue damage by cytokines, so we will talk about it a bit later. And then more phagocytes can be recruited from the bloodstream, so there can be a really big accumulation of these cell types. And when they are activated, they release cytokines. And some of them, as I told you, not the neutrophils, but the dendritic cells and the macrophages, they also can present antigens to cells of the adaptive immune system. So if you look a bit more into detail about the different phases that you can distinguish in the process of phagocytosis, you can see that this is a phagocyte, and this is something that has to be phagocyte host. For example, microbe, it can be a particle or cellular debris, as I said before. Then this phagocyte extends its cell membrane, so it forms so-called pseudopodia, and these really engulf the thing that has to be taken up. Then a so-called phagosome is formed, and this phagosome you can just see as a vesicle which has been which originates from really the cell, so this part of the outside world, so to say, still, and which has really been internalized. Yes. That it's a microbe. I will talk about it in the second part. So the recognition systems, there are really many different kinds of recognition systems, and I will talk about it a lot later. So this phagosome contains the thing that should be cleared, and to do so, it first needs to be broken down, and this is being done intracellularly, because the phagosome fuses with a so-called lysosome, which is a cell organelle, and which contains a lot of digestive enzymes and also a very low pH. And together, this aggressive milieu makes sure that anything that has been internalized is being broken down in so-called phagolysosomes. So in this phagolysosome, digestion takes place, and in this way, of course, also the microbe or anything else is killed and is being cleared. And sometimes a little bit of indigestible material still remains, and this is just released by the cell again. As I said, in this process, if this occurs in a macrophage, then parts of this microbe or whatever has been taken up is also presented, but it doesn't always have to be. So in this case, this is a neutrophil, for example, and this just makes sure that it's really being cleared. So the neutrophils are the most important type of phagocyte. Most of the cells in the blood are neutrophils, so about 60 to 70% of all leukocytes. As I said before, contain a lot of granules and have this really characteristic shape of their nucleus. You can see it here as well. Lots of segments. Normally, if there is no infection going on, you should not see them in tissues. So they are only in the circulation, and only when there is tissue damage or infection, then you can also find them in tissues. They have a very short lifespan. It's not really clear if it's really 12 hours, but it's in this area, so 12 hours, a little bit more, a little bit less, but very, very short-lived cells. And as I said, these are really the first cells to respond to an infection or injury at an inflammatory site. They then ingest and they kill the pathogens or the dead cells or anything that has to be cleared. And in this process, and during this process, they release lots of different proteins that are either cytotoxic or that are helping to generate an inflammatory response, these cytokines. And lots of these compounds that are secreted are present in their granules. Can be also proteolytic enzymes, so enzymes that really break down different proteins. Can also be a different class of cytokines called the chemokines, which are important for attracting other inflammatory cells. And they also make a lot of reactive oxygen species. So they have really multiple actions to efficiently clear any danger, and there you can really see them as a kind of suicide cell. So they really are ready to go. It's a bomb full of different really toxic substances. So, for example, well, in these granules, lots of the primary granules you can distinguish, and the specific and tertiary granules. There are different kinds of proteins present. For example, elastase, which breaks down extracellular matrix proteins like elastin, but also a protein called myeloperoxidase, which is really maybe one of the most important effector molecules of the neutrophil. So if you think of an infection and you see pus, for example, you see it's a bit green yellowish. This color is all due to this myeloperoxidase protein, because it contains a heme group, and this really gives that color. So this is all related to this protein. And this protein works together with another protein on the neutrophil, which is called phagocyte oxidase. And this protein generates lots of different reactive oxygen species. So, for example, the hydroxyl radical, but also it generates hydrogen peroxide. And this hydrogen peroxide is being used by the myeloperoxidase to make various different compounds. For example, hypochlorite, which is indicated here. And this hypochlorite is, for example, the thing that you use to clean your toilet. So the bleach that you use to clean is really this hypochlorite, and this is really also produced inside your own body by your own cells. So you can imagine this is really an aggressive substance which really kills pathogens. Then in these specific granules, lots of antimicrobial proteins. For example, this lysozyme and lactoferrin again, I referred to before, they are present there. They are secreted also by the neutrophils. Then some antibacterial proteins like calprotectin, they're still present in the cytoplasm. So also the cytoplasm contains harmful substances for pathogens or cells that have to be cleared. And then quite recently, it was discovered that even the nucleus is being used by neutrophils. So when their life cycle ends and they have released everything that they have to kill a pathogen, they can still use their DNA, and they do that with forming nets that really trap bacteria. This is really, I think, a nice function. This shows how it works. So if it really is down and out, so to say, it still can release its nucleus. And here in red you can see the DNA, and it's really being expelled. And in this way, it can trap bacteria. So I think this is really a cool function also of neutrophils. Then the macrophages. Macrophages are being formed after differentiation of monocytes from the blood. Monocytes make up like 1 to 6% of all the leukocytes. I know the reference ranges are a bit unclear sometimes, but this is the amount that you may expect in the blood. These monocytes are produced in bone marrow, and they may enter the circulation and then migrate to inflammatory sites. And when there's an inflammation going on, and only then, they differentiate into macrophages usually. So as I said before, when there's inflammation, first the neutrophils arrive, they are ready to go, so they make sure that the first phase of the protection takes place, and then macrophages arrive later, from 3 to 7 days after the neutrophils. And usually they are not present in tissues, so they are also, if you encounter them, it might be viewed as a sign of inflammation. But in some tissue types, they are also resident, so they can be present always, also when there is no inflammation. And this, for example, is happening in the gastrointestinal tract, in the lung and in the liver. For example, the Kupffer cells you've been studying before, they are macrophages which are always there, also if there is no inflammation. And this usually, these macrophages that are resident, they are present there because they encounter a lot of pathogens anyway, because they are close to the outside world. The liver macrophages, for example, so the Kupffer cells, they of course, if there's translocation of bacteria from the intestine, they first are seen by the liver. So the Kupffer cells in the liver make sure that they are cleared. And the same goes, of course, for example, the lungs. Here? These? Oh, the resident cells. They are. You can see them as macrophages. There are no monocytes anymore. Yeah. So, and when they become activated, these macrophages, they really become active. They increase their size. They increase the number of lysosomes. These are needed to fuse with the phagosomes, which to make sure that phagocytosis occurs right. And they also make a lot of secretory products. I will talk about those later. And you can see here that they really look different, a monocyte and a macrophage. Yeah, I think I've seen this before. Then cytokines, I've talked about them before many times now. These are proteins mostly, yeah, sometimes very short proteins, peptides, that are being used for communication between immune cells, but also between immune cells and other cell types. These cytokines usually bind to receptors, so cytokine receptors, which are present on the surface of cells, and they induce changes in those cells. And it can be many different actions they have. They can induce growth, but they can also induce death. They can induce differentiation of cells. They can induce movement. So there can be many different responses depending on what type of cytokine is secreted. And then there's many different kinds of cytokines. So you can broadly divide them into 4 classes. The first one is the interleukins. These interleukins are really also a big family. It's like interleukin 1 to somewhere in 50. So many different proteins. So they have also various functions, and they basically talk to also the adaptive immune system, but they also make sure that cells of the innate immune system can talk to each other. And there's a prototype pro-inflammatory interleukins like interleukin 1 and interleukin 6, but there's also interleukins that have more an anti-inflammatory role, which is maybe important for wound healing responses or delaying or resolving inflammation. This is, for example, a function of interleukin 10. Then there's another class. Second class is called chemokines, and these chemokines are very important for promoting migration of cells to sites where they are needed. So I will talk about it later. They are important for the process of chemotaxis. Then there are tumor necrosis factors, which the name says it a bit already. They help specifically to kill tumor cells because they initiate apoptosis, but they can also initiate apoptosis in cells that have been infected, for example. So tumor necrosis factors are important for that process. And interferons are mainly known for their function in the control of viral infections. These proteins, they activate natural killer cells and macrophages. They can also be made by these cell types. And they promote the recognition of tumor cells by these cell types. Maybe we have to stop a bit, or you need a break, or still okay? Should I go on, or no response? All blunted. You want to break, or you want to go on? Yeah, okay. So let's break for 10 minutes, maybe. Um, so in this part, I'll talk mostly about the more molecular mechanisms that are involved in chemotaxis, but also in recognition of the different types of dangers. And first I'll talk about this chemotaxis. So I think there was one question before, how these cells know where to go. And this is being done by a process called chemotaxis. And how does that work? Well, if there is an inflammatory site, for example here, there's a very high concentration of bacteria present, or in this case bacteria can also be viruses, of course, or just an injury. And immune cells, they sense this because there's a very high concentration of either bacterial products or specific cytokines called chemokines, for example interleukin 8 or monocyte chemoattractant protein 1. This interleukin 8 is very important for the recognition of neutrophils, and MCP1 is important for the recognition of monocytes. These have a very high concentration here because they are being secreted by the cells that have been infected or damaged. And also complement fragments can be generated at the site of inflammation. And there's a very high concentration here, but the further you go from the site, the lower the concentration is. And for example, here a macrophage senses a specific kind of chemotactic signal by a receptor on its cell membrane, and then it just moves into the direction of the higher concentration. So this is the chemotaxis process, which is mediated mostly by chemokines. And how does that work? I think this has also been covered already, probably by the introductory lectures, so I will go over quite briefly. So there's different phases, rolling of the cell when it's slowing down over the endothelium, still in the bloodstream. Activation of the cell when there is a more firm adhesion between the endothelial cell and the immune cell. Then it can really stop the arrest or adhesion phase, and finally it can perform transendothelial migration. And you can see a picture of that here. So this is the blood vessel, and there's lots of immune cells stopped on the endothelial cells of this vessel. If you go a bit more into detail, you can see that various adhesion molecules are involved, are important. These adhesion molecules are upregulated by the endothelial cells. They are being presented on their cell membrane because these endothelial cells are activated by mainly tumor necrosis factor alpha and interleukin 1, which again originate from macrophages that have already initiated the inflammatory response. So these adhesion molecules are on the cell membrane. A leukocyte, in this case a neutrophil, has different kinds of receptors on its cell membrane. And first there's an interaction between selectins and the selectin receptors, so the selectin ligands and selectin receptors on the endothelium. The leukocyte, the neutrophil, gets activated. There's also an interaction between the chemokine receptor on the neutrophil in this case, and the chemokine itself, which is present on the surface of the endothelial cells. It's coupled to proteoglycans, so to glycoproteins. And then there's a third interaction needed to establish a really firm adhesion, and this is an interaction between integrins on both the endothelial cells and the neutrophil, and then you get really a firm adhesion. And in this phase is called margination also, or pavementing. And then there's this transmigration I told you. This is also referred to as diapedesis. So it moves in between two endothelial cells, and then by this process of chemotaxis it moves to the site of the inflammation and helps to clear the problem. So which sensor systems are in place to detect all these dangers? There's many different types. And the main thing that you should really remember is that they respond to patterns that are associated with danger. And they don't respond to molecules specific to a given pathogen or a danger. This is in contrast to cells of the adaptive immune system, which really recognize very specifically only to a very specific part of a danger, of a pathogen or a cell that has been transformed, or a protein. The cells and the molecules that are part of the innate immune system, they only respond to patterns that are characteristic of many pathogens or many dangers. And these are called pathogen-associated molecular patterns, or PAMPs, or danger-associated molecular patterns, DAMPs. And the structures that can be recognized, it can be many different things. It can be sugars, specific types of sugars, can be proteins or nucleic acids. We will see later a bit more specific with what kind of molecules these can be. So you can distinguish three important groups that take care of the detection of danger. First group is the toll-like receptors and the NOT proteins. They belong together. Then there are the inflammasomes. And also the complement system is important for this. And all these receptor systems, they are so-called pattern recognition receptors. So they recognize these patterns that are associated with danger. And these sensors allow the cells of the immune system to see molecules that signify the presence of danger. And for example, here this is a bacteria, the E. coli bacteria, which is a gram-negative bacteria. If you look at its cell wall, then you can see that this peptidoglycan is present, but since this is a gram-negative bacteria, it also has lipopolysaccharide or endotoxin on its outer membrane. And these molecules can be used by the innate immune system to recognize these bacteria. And basically all gram-negative bacteria can be recognized by sensors that detect this lipopolysaccharide, because this molecule is only present on gram-negative bacteria. So if you look at toll-like receptors and NOT proteins, then toll-like receptors have been discovered like maybe 15 years ago only, so there's only quite recent data. And these toll-like receptors are transmembrane receptors that recognize parts of pathogen products that are present in different types of pathogens. Then the NOT proteins basically look the same, but they are present intracellularly, and therefore they recognize microbial material that has already entered the cytoplasm of the cell. So it's not on a membrane, but in the cell, inside the cell. Most of the cell types that are present in our body express these toll-like receptors and these NOT proteins. So most of the cells are equipped to sense the danger. And then when these molecules, these TLRs and these NOT receptors have encountered a danger signal, then there's a binding, of course, between these danger signals and the receptor itself. And a whole signal transduction cascade is activated. Signals are transmitted usually in the cytoplasm, but also to the nucleus of the cells. And these cells that have been infected or have encountered danger, they generate a pro-inflammatory response, and they synthesize by gene transcription and translation a lot of different molecules that are important for mounting a good inflammatory response. For example, this interleukin-1, but also interleukin-8, interleukin-6, but also these various defensins that we have been talking before. Their release and their secretion and production is all increased after binding of these toll-like receptors to dangers. So what kind of compounds are recognized by these toll-like receptors? Can be many different things. For example, I told you before about the detection of lipopolysaccharide on the outer membrane of gram-negatives. This is being recognized by the toll-like receptor 4, but also flagellin, for example, which is part of the tail of some kinds of bacteria, the flagell. They are being recognized by TLR5. Then there's also different types of danger signals that are associated with viruses that can be recognized by toll-like receptors. For example, single-stranded RNA and double-stranded RNA. Specific types of these RNA molecules can be recognized by different types of toll-like receptors. So these are examples of molecules that are recognized by toll-like receptors. Sorry. Mm-hmm. NF-kappa B is a very important signal transduction molecule which has various pro-inflammatory roles, and it basically is just an intermediate between the TLR and the gene transcription. So it is more or less a transcription factor, and it's usually inhibited by this I-kappa B, which is also on the slide. And if this I-kappa B is released, the NF-kappa B becomes free to act really as a transcription factor. It translocates to the nucleus, and it promotes the transcription of these compounds that are important for the inflammation. So it's a transcription factor which is really intermediate between TLRs and the production of molecules important for inflammation. And most of the pro-inflammatory responses are characterized by activation of this NF-kappa B. It's really a central molecule. Then, apart from the toll-like receptors and the NOT proteins, there's a different type of sensor system which has really only been discovered like 5 years ago, which is called the inflammasome. And the inflammasome senses other types of dangers. For example, it senses extracellular ATP. You know, ATP is an important molecule for energy. How do you say? It's the energy carrier of the body, which is usually present in cells, but not outside of cells. So when cells leak ATP, it signifies that there is a problem with the integrity of the cell, and this should be picked up by in some way by a sensor system. In this case, inflammasome proteins, and also other danger signals like crystals of cholesterol and urea, which may puncture cells and in this way also damage them. Or bacterial toxins, and again, cytosolic flagellin can be sensed by the inflammasome proteins. And the inflammasome itself is a multi-protein complex. It consists of caspase 1 and ASC usually, and then one of the other proteins that mediates the recognition of these specific signals. For example, NLRP3 is one of those important proteins. And together this assembles, and if it gets activated by a signal, then caspase 1 becomes activated, and this caspase 1 molecule is capable of activating in turn cytokines like interleukin 1 and interleukin 18. So only when there's active caspase 1, these molecules are matured, and they can be secreted and then exert their inflammatory function. So this is another important sensor system. And then the final sensor system, this is the complement system. I will talk about it a bit more in detail. The complement system is a really old system, so like what I told you before, the cell types and so on. Also, this may be one of the most conserved parts of the innate immune system in all organisms. There are many proteins that are part of this complement system, but there's like 20 core proteins that are important. And this complement system, it belongs to the innate immune system, but simultaneously it also acts like a bridge between the innate and the adaptive immune system. So most of these complement proteins are secreted and synthesized by the liver, but they end up in the blood plasma, so they circulate in the blood, but they circulate there in an inactive form, so they are not active. And they have been named, most of them have been named conveniently by number. So from C1 to C9, these molecules are part of the classical pathway of complement. I will talk about it later more in detail. Factor B, for example, is also an important protein which is more involved in the alternative pathway of complement activation. These complement proteins, some of them act as sensors. They detect the presence of foreign material. It says here it can be pathogens, it can also be cells again that have been injured or transformed or malignant. And then a cascade of reactions follows. And in this cascade of reactions, one complement protein activates another one in the cascade, and this protein activates yet another one downstream. And then in the end you get a real big amplification of the whole activation system. this is what I said before. So it cooperates with other defense systems. It can also, we will talk about it later, but it can also mediate phagocytosis, and it can stimulate the adaptive immune system. Yes? Sorry? Where? The cell or the proteins of the complement system are mostly synthesized in the liver. But there's, I mean, it's not exclusively, but the majority of them are predominantly secreted by the liver. But also, like the intestine can make also a bit of C3, for example. C1q, which features in the classical pathway, is also present in monocytes, for example. So, but the bulk comes from the liver. And then there's different kinds in which the complement system can be activated. So there's 3 pathways basically, and these are referred to as the lectin pathway, the classical pathway, and the alternative pathway. So the lectin pathway features molecules which are specific to this lectin pathway, like MBL, so this stands for mannose binding lectin, and also phycolins. Then the classical pathway becomes activated or is dependent on the recognition of antibodies by complement factors, and also pentraxins can be recognized by complement proteins. And then alternative pathway activation occurs directly on microbial membranes, or also sometimes on our own cell membranes if appropriate. And then all these pathways basically they end up in a common pathway after some early events. And this common pathway, and the thing that is really common to all pathways, is the cleavage of the central complement component, which is C3, by a protein complex called the C3 convertase. So this C3 convertase is always formed in both after activation of lectin, classical and alternative pathway, and this mediates the cleavage of C3. And the C3 convertase enzyme itself is the same for lectin and classical pathway, but it's a different kind of convertase for the alternative pathway. But the thing that they have in common is that they always make sure that C3 is activated and becomes active and is cleaved, because that really is what the activation is. So it's always cleavage of a molecule. And then after that, there are various late events that can occur. So if the complement cascade just follows further, then other complement complexes are generated, which in the end together form a so-called membrane attack complex. Some parts that are generated promote inflammation and promote chemoattraction of cells. Specifically C3a and C5a are important for that. Then C3b is important to induce phagocytosis, and it's also involved in the promotion of antibody production. So this pathway a bit more in detail is depicted here. So this is called the terminal pathway, and it's still part of the complement system, and this terminal pathway results in the formation of the so-called membrane attack complex, which forms holes in bacteria and thereby kills them. So in the first step, after the C3 has been activated, a so-called C5 convertase is formed. And the C5 convertase, which is a very difficult molecule here, makes sure that C5 is cleaved. So a big fragment is generated, C5b, and a small fragment, C5a. And the C5b then recruits other complement proteins, so C6 and 7 and C8. And in the end, various subunits of C9. And together, these C9 subunits, they really form holes, as you can see here, and they drill holes in bacteria. And the components that are present in the cell leak out, and this really effectively kills bacteria. But it can also kill our own cells of our own body. So we need to be protected from this complement attack. And actually there's a lot of inhibitory proteins that are being synthesized by our own cells to prevent our attack by our own complement system. So if we look at the classical pathway and how this is initiated, I told you before that this is dependent on antibodies. So when an antibody recognizes an antigen, it can bind, of course, and the antibodies, and then specifically the constant part of these antibodies, this can interact with the first complement protein, so C1, and then specifically the C1q subunit. And you can see it here. So this is a bacteria. There's different antibodies that have recognized this bacteria. This antibody then is recognized by the C1q subunit, and then there's different other subunits of this C1 recruited, and together they make sure that C2 and C4 are cleaved. And when C2 and C4 are cleaved, then there's this C3 convertase, which is C4. This is not right. C4A2B. C4. Wait, sorry. When the it's okay. The C4B2A is formed, and this C4B2A is the C3 convertase, and this makes sure that C3 is activated. Is it clear? You need to know the steps of the complement cascade. Yeah, maybe it's a bit more molecular now. Yes. How they are separated? It's one molecule for C2, but after cleavage, so C1, if it gets activated, it becomes proteolytic, so it can cleave the second complement factor, and the cleavage makes sure that C2A and C2B are formed. Does that answer your question? Yeah. And then for the lectin pathway, it looks very much like the classical pathway. But it's not dependent on C1q, but on a molecule called MBL, mannose-binding lectin. And this mannose-binding lectin, it does not recognize antibodies, but it recognizes mannose subunits. And these mannose subunits, or mannan it's also called, is a type of sugar that's only present on microorganisms, so on bacteria and yeast, for example. But we don't have it. So this is again a pattern that can be recognized. So if this mannan or mannose is present, then our MBL protein, which is also again synthesized in the liver, binds to this mannan, and then other molecules like MASPs are generated or are recruited, and together they also again make sure that C4b and C2a are formed, and then this is again the C3 convertase, which makes sure that C3 is converted and becomes active. And the alternative pathway is quite different. So there's no C4b or C2 or C1 involved there. It's different proteins like factor B, D, and P here. And the alternative pathway is really a more an amplification pathway. So a little C3b is always present. It's always a little bit activated. This process is called tick over activation. And this C3b, if it forms a complex with a factor B in this case, this is another type of C3 convertase. So together, this can amplify and generate more C3. So the C3 that has been formed in the alternative pathway can promote its own formation again and thereby amplify the complement cascade. So this, of course, again is very dangerous also for our own cells, because if there's a little bit C3 activated, C3b formed on our own cells, you might also attack your own cells. So this, again, is very tightly inhibited by various regulatory proteins.
I have a question. Why do they call it C3b?
Here. This is a complex of C3b, just like the normal C3b that has been generated after cleavage of C3. And this is a complex with a so-called activated factor B. So then they call it BB again. So this is just factor B which has been activated. Yeah, so this is C3b, the C3b which is generated after cleavage of C3. So this is part of C3, and then factor B which comes in here. Yes. And the complex is again a C3 convertase. It's quite complicated, maybe, yes. Anything unclear about this? Yes. This can also activate the C5 again. So this can act as a C5 convertase. Yeah. So in all pathways basically can initiate the activation of the terminal pathway also. Yeah. So then to summarize and make the bridge to the adaptive immune system, the differences between innate and adaptive immunity. So I told you that the innate immune system acts really quickly, in minutes to hours, whereas adaptive immunity takes much longer to evolve and takes days. Innate immune system is really dependent on the recognition of molecular patterns which are associated with pathogens. And in adaptive immunity, no patterns are recognized, but really specific molecules and even parts of molecules only. But a very limited number of receptors are important for the recognition. Just the toll-like receptors, there's not a lot, there's only 9. The different inflammasomes and the different complement sensors. And in the adaptive immune system, there's a lot of receptor genes, all the types of T-cell receptors and B-cell receptors that can be present. There's many different ones. There's no memory in innate immunity, whereas in adaptive immunity, there's a very important memory response. Then, because innate immunity relies on really patterns that are only present in cells or things outside of our body, there's a really good discrimination between self and non-self, and you know that in adaptive immunity autoimmune diseases can also develop. So in this case sometimes there's a problem here in adaptive immunity, but this does not occur in innate immunity. There's a lot of antimicrobial peptides and proteins that are acting as the soluble components of the innate immune system, and this task is performed by antibodies in the adaptive immune system. And I showed you the major cell types like the phagocytes and also the natural killer and dendritic cells that are part of the innate immune system, whereas in the adaptive immune system it's the T and B cells that are really the main cell types there. So this is what I would like to say. I don't know if you have questions.
So do we have to know the way of the cascade in all 3 pathways?
Maybe not in as much detail as here, but I think it was for this case already, right? The complement system. I don't know the level that they provided there. You should be, or that you did see in the textbook. Is it very different?
And normally they say that the lectures are
Okay. Oh, then it's clear. Huh? But actually it's not so difficult. I mean, alternative and lectin, or classical and lectin basically are more or less the same, apart from the classical pathway being dependent on C1 and the lectin pathway on MBL. And then they both make sure that C4 and C2 are activated. And then C3 I mean, it's, yeah. The only thing that is counterintuitive is that C4 is before C3. This is the only thing you need really to remember, I think, which is not logical. Not from a book. They are from different papers in journals. Yeah. I still have to add the sources, you're right. Usually macrophages can do it repeatedly, so they can take up a lot of different pathogens, bacteria or cells that have to be cleared. Before, if they really end their life cycle, they just go into apoptosis, and they are being phagocytosed themselves by other macrophages. So they become, this is also one of the reasons why they become bigger when they get activated, because they just keep on engulfing and phagocytosing stuff. And then in the end they die by apoptosis, and they are also being removed by other phagocytes. Can also be neutrophils, but mostly macrophages. Where? At the site of the inflammation. They are there's a whole different process going on then. For example, there's a lot of autophagy going on, so it's basically a recycling of cell components, because of course if everything is being digested, you get the building blocks of a cell again, and so you can reuse it, and it is being reused for new cells. So it releases also nutrients. Yeah.
After the macrophage or neutrophil phagocytose a microbe, does it do anything? I think it sends cytokines.
Yes, it makes cytokines. Yeah.
Are those always the same, or does that depend on?
No, it's not always the same. There's if you look at macrophages, there's very many different types of macrophages, subtypes. This can really make a big difference for what kind of cytokines they secrete and what kind of role they play in the inflammation. So there's a basic distinction between so-called M1 and M2 macrophages, in which the M1 macrophages are really pro-inflammatory. They make a lot of pro-inflammatory cytokines like IL-1 and IL-6, IL-8. And then M2 macrophages are so-called regenerative macrophages. They are more important for wound healing and remodeling of tissues. And they make more interleukin 10 and arginase and proteins like that. So there can be many different more layers added to all these cell types, and there's much more to be known. But I guess that's for later years. Okay. No more questions? Good luck then.